Hello and welcome to our module entitled the parasitology focused on protozoans. Okay, so for the learning outcomes of today's topic, number one is for you to understand the basic concepts of protozoan life cycle and pathogenesis. Number two, recall the significant features of different protozoan parasites that aids in the laboratory diagnosis. General characteristics of protozoan parasites. Generally, the protozoan parasites are unicellular organisms, meaning they only contain a single cell for that particular organism. And they don't have any cell wall, but they have what we call the periplast. So, periplast is an example of a plasma membrane wherein the nutrients as well as the waste products of your protozoan parasite will be absorbed and released there. No? So that is the function of their periplast. Another thing about the protozoan parasites is that they have two regions of cytoplasm. The first one is the endoplasm, which is from the word endo, syempre, it is inner, while the ectoplasm is in the outer. So in the ectoplasm, you'll find there the locomotory organelle or the organelle for their locomotive responses, no? Meaning this is the organelle for their movement. Okay, so later on we're going to discuss about that as well. Okay, so they can asexually reproduce. So they have the trophozoites and cyst forming. Other protozoan parasites such as apicomplexans can reproduce sexually, no? And then later on we're going to discuss about that. So trophozoites can reproduce through the binary fission, while the cyst forming can reproduce through nuclear division. So vacuoles for storage and transport. So meaning all of the protozoan parasites contains vacuoles. They are like small opening, no? Parang ganun yung itsura niya sa mga cells. And then that is for their storage and transport. We can classify our protozoans according to their locomotory organelle. So we have 4 different classes and subphylum for the protozoans. The first one is the Sarcodina or the Rhizopoda, and the locomotory organelle present on the ectoplasm of those protozoan parasites is what we call the pseudopods. So let me draw, no? So pseudopods are like finger-like structures, no, present in their ectoplasm. So you try to imagine yung kamay nyo, no, ilapag nyo sa something sa surface, no, and then try nyo yung kunwaring umaastang gumagapang. So parang ganun yung locomotory organelle na to, no. It's a finger-like organelle, no. The next one is the Mastigophora or the Flagellata. So in other words, they contain flagella. So these are the Flagellata. Yeah. It's either they contain 1 or 2 or 3, no, flagella. The next one is the Ciliophora or the Ciliata, meaning they have cilia or hair-like structures all throughout their cell membrane. And the last one are apicomplexans, and they are indefinite. So the apicomplexans, they have these sporozoans and coccygians. And from the word itself, apicomplexans, meaning they are inside something, no? So they require another cell for them to be transported. Okay, so another cell. So they don't have any locomotory organelle. Instead, they require another cell or a vector, no, for them to travel or to move. Okay. Furthermore, other than the locomotory organelle, we can also classify protozoans according to their taxonomy. So once again, we also have four. No? The first one is the subphylum Sarcodina, superclass Rhizophoda, and class Lobosea. Under this subphylum, the members are Acanthamoeba castellani, Endolimax nina, Entamoeba coli, Entamoeba dispar, Entamoeba gingivalis, Entamoeba histolytica, which is pathogenic, the only pathogenic pseudopod, no? Iodoamoeba bushley, and Naegleria fowleri. So the only pathogenic here is the Entamoeba histolytica. However, we have also opportunistic amoeba here, no? So they can be pathogenic when the situation arises. So we have the Acanthamoeba and Naegleria. The other parasites are called commensals. So later on we are going to discuss about them. Next, under the phylum Protozoa, we have the class Flagellata. In the atrial flagellates, we have the Chylomastix mesnili, the Entamoeba fragilis, Jarjolamnia, Trichomonas hominis, Trichomonas tenax, Trichomonas vaginalis. And for the hemoflagellates, we have the Leishmania species. Under that, we have the Braziliensis, Donovanian tropica, as well as the Trypanosoma species. We have the Brucei complex, and Cruzi. Next, under the Ciliata, we have the only one significant parasite. No? It is the Balantidium coli. And for the last class under the phylum Protozoa, we have the class Sporozoa and the class Coccygians. Under the class Sporozoa, we have the Babesia or the Babsia species, Plasmodium species. Under the Coccygian, we have the Cyclospora species and the Toxoplasma gondii. Okay. So first topic is the subphylum Sarcodina superclass Rhizophoda, or the pseudopods. Before we go to the topic of the pseudopods, we need first to understand the life cycle behind them. Though they have four major stages, only two are significant. And sigurado alam nyo na yon. Sino? Si cystic stage and si trophozoite. No? So we have the cystic stage and the trophozoite. So almost every amoeba that you're going to encounter have the cystic stage and the trophozoite stage. Okay? So the cystic stage, it is your non-feeding stage, while the trophozoite is your feeding stage. Okay? So first one is rhizopods. Their locomotory organelle is the pseudopodia. The life cycle involves the trophozoite, the pre-cystic stage, cystic stage, and the metacystic stage. And please take note of this. All of amoeba has trophozoite and cystic stage. Only one does not have the cystic stage, and that is Entamoeba gingivalis. And another thing, all of them, all of the amoeba has the large intestine as their habitat, except for Entamoeba gingivalis. Because Entamoeba gingivalis, the habitat is the oral cavity. Next, all of your amoeba are all commensals, except for Entamoeba histolytica. By commensals, what do you mean? What do you understand? What's the definition of commensals? So commensals are your parasites wherein they gain benefit to their host. However, they neither cause any harm or any damage on that particular host. So basically, they are free living on their host and they don't give any pathologic conditions. Unless, syempre, there is a particular problem. No? So commensals are on your host. Okay. Next, rhizopods, we have the entamoeba, meaning these are your true amoeba because they have the peripheral chromatin and chromatoidal bodies in their cyst. Okay, please take note of that. All of entamoeba species are true amoeba because they have the PC and the CB. And so, if all of the entamoeba species are true amoeba, paano naman sina Endolimax and the Eudamiba? So they are the other amoeba because they don't have the peripheral chromatin and the chromatoidal bodies in their cyst. So the infective stage is cyst, while the mode of transmission is ingestion of cyst. So to give you a differentiation of the trophozoite versus the cyst, okay, please look at this table. Number 1, motility. Trophozoites are highly motile, and the presence of the pseudopodia is very much seen in this stage. While in cyst, they are non-motile. No? They are just the cystic stage, meaning they are circle, no? Parang ganun lang, spherical in nature. Another difference, the trophozoite is the feeding stage of your pseudopodia, while the cystic stage is the non-feeding stage. So trophozoite is the vegetative stage, while the cyst is your infective stage. Trophozoite is present in diarrheal and liquid stools, while the cyst in the formed stools. So basically, trophozoites should be assessed within 30 minutes. So if you see diarrheal, the urate or the liquid stool in your specimen, and if you are trying to rule out trophozoite, kailangan you need to perform or conduct the fecalysis within the 30-minute mark. Okay, after that, syempre, ma-destroy na or masisira na yung mga trophozoites mo. Okay. So we have the pathogenic amoeba, the only pathogenic amoeba, which is your Entamoeba histolytica. The Entamoeba histolytica can cause the disease amoebic colitis as well as the ALA, or the amoebic liver abscess. So why do this particular parasite can cause such abscess? It is due to the three virulence factors. Number one, lectin, amoeba-4, and the cystine proteinases. So it uses its chemical compound lectin for adherence, meaning for that particular parasite to attach on the tissue cells na it likes to ano infect, no? Nahirapan ako mag-English doon, sorry. And then amoeba-4, on the other hand, the purpose of the amoeba-4 is to produce spores into the cell, meaning bubutasin niya, no? So why do that bacteria ako na. Why does this parasite want to produce such spores? Kasi it will introduce the cystine proteinases. It is an enzyme. It is a enzyme that wants to destroy, no, the cell itself, no, yung mga tissues. So it is a cytopathic enzyme. Okay, so remember these three, no, the three virulence factors of your Entamoeba histolytica. We have the lectin for adherence, amoeba-4 to produce such pores into the cell, cystine proteinases, an enzyme, and then cytopathic enzyme. So as a distinguishing characteristic, your Entamoeba histolytica has a trophozoite which is moves in one direction, which is progressive. It contains one pseudopod, a central chirosome, and a fine chromatoidal bodies. While the cystic stage, it has a 4 nuclei that is coffin shape. So these are the other amoeba. They are commensals, or the non-pathogenic amoeba. The Entamoeba coli, which has a very much similar with Entamoeba histolytica, a bit bigger and has more nuclei present in their cyst, no? And the difference pa is the trophozoite, no? This is a major difference between the two. The trophozoite of Entamoeba coli it has the several red direction, unlike the histolytica, which is a single direction and progressive. So Entamoeba coli is several direction and sluggish. It has several pseudopods, eccentric cardiosom, and uneven chromatin. So the other commensals, we have the dispar, is morphologically similar to histolytica but different DNA and RNA structure. And Entamoeba hartmanni, it is similar with the histolytica but a bit smaller. Endolimax nina is the smallest amoeba. Entamoeba pollecki is the amoeba of your monkey and pigs. Entamoeba gingivalis has no cystic stage and is in your oral cavity. And the Iodamoeba bushleyi is uninucleated and stains deeply with iodine from its name itself, no? Iodamoeba, so it stains with iodine. Okay? And we also have your opportunistic amoeba. Dalawa lang naman yan. So they are free-living amoeba that inhabits the central nervous system. So we have two, the Naegleria fowleri and the Acanthamoeba castellani. Once again, they are called opportunistic amoeba kasi they're only pathogenic if the situation is good for them, meaning accidentally they can go through your eyes, ganon, or your nasopharyngeal area. Okay, although they are free living, meaning they don't need a host for them to to do their life cycle. No? So, free living sila. So, Naegleria fowleri is the major causative agent of PAME. What is PAME? It is the primary amoebic meningoencephalitis. Naku, okay. So, it is the primary amoebic meningoencephalitis. No? It could cause purulent spinal fluid. Do you mean by purulent? Meaning it is full of pus, no? Excuse. It's full of pus. And the pus are WBC, and in particular, if this is a parasite, the usual WBC should be eosinophil. However, in this meningoencephalitis, it is not eosinophil. Please remember that. It is full of neutrophils. Okay, kindly take note of that, no? Even though this is a parasite, kasi it is a unicellular in origin, no? So the body will detect it as a bacteria. So basically, instead of eosinophil, our body will respond, or our immune system rather, will respond with neutrophils, no? So it is the primary amoebic meningoencephalitis. So it is acquired through swimming during hot weather in ponds, no? Sa mga ganong area. The next one is the Acanthamoeba castellani, and it could cause the keratitis. On chronic Acanthamoeba infections, it could cause the GAE, and GAE is granulomatous amoebic. You can spell amoebic with ganito, no? Or amoebic. It's up to you now. It's the same lang naman. Encephalitis. Okay. So, granulomatous amoebic encephalitis. The difference between the primary meningoencephalitis, the Naegleria fowleri will damage your meninges, no, yung thin covering ng brain and ng spinal cord. Pag naman GAE or granulomatous amoebic encephalitis, in your yung covering ng brain mo, ang gagawin nila is magpapadala yung inyong immune system ng maraming WBC doon sa parasite na nandoon. So, example, andito yung utak mo, no? Yan, ito yung utak mo kunwari. Yan. Yan. Okay, yan yung utak mo. Utak yan, ha? And then, nandito yung parasite. So, magpapadala. Yan, gawin natin red yung parasite. Yan. So, magpapadala ng WBC yung inyong immune system to combat that parasite. However, ma-stuck sila doon, yung mga WBC. They can't go back to the bloodstream. And then, what will happen? It will form a granuloma. And granuloma is the parang bilog, no? Yan, parang collection ng bilog na WBC. Yan, granuloma. Kaya siya tinawag na granulomatous amoebic encephalitis. Next is the class Mastigophora, or the flagellates, and the hemoflagellates. For the next one is the flagellates.